probably seen it countless times in TV crime shows. The killer was careful. He wore gloves so he wouldn't leave fingerprints, and he covered his tracks to confound police. But then, what's this? A hair that was left behind? Quick, send it to the crime lab. And sure enough, through careful DNA analysis by a team of forensic specialists, the killer is identified and caught.
How many people have ever seen some of those crime-solving shows where they do this thing called DNA fingerprinting? Yeah. What is that?
That's Homer High School science teacher Vicki Lowe, who knows a thing or two about DNA. She's speaking to Mrs. Gribble's classroom full of sixth graders at West Homer Elementary, who are in the middle of a special lesson on DNA. At the end of the lesson, they may not catch a killer, but they'll have something just as cool, a visible sample of their own DNA that they can take home and show their parents. Lowe says the first step for the students was to collect a self-DNA sample. That's done by gathering cells from inside their cheeks.
So they chewed on the inside of their little cheeks for 30 seconds, and then they swished with some plain drinking water, and they put the swish into a cup. After that, they added a chemical to break open the membranes, and then they treated their sample with an enzyme that broke up proteins that the DNA was wound to.
The enzyme is called protease, and when it's mixed with a little bit of salt solution, it helps clump the DNA together into a form that can be seen with the naked eye. The next step is to gently rock the test tubes back and forth to mix the solution.
Hold it like this. 1, 2, 3, 4, 5. So we've mixed it, but it shouldn't be super sudsy.
The kids are grouped together at tables in teams of 5 or 6. At each table is one of Lowe's students from Homer High School, lending a hand in making sure the procedures are followed correctly.
All right, so what do you guys see now?
No, this is from the detergent. Yeah.
What do you think that is?
Maggie Graham is a high school senior. She's guiding her kids over to a main table in the front of the classroom where they each place their test tubes in a small pool of hot water. What does this thing do?
The water bath, it just it makes the separation more definitive, and it brings the DNA to the top and the water to the bottom, and the protease and lysis buffer.
And it has to be in there for 10 minutes?
Mm-hmm. Yeah.
After the water bath, the last step is to add some really cold alcohol and then let the concoction sit for about 5 minutes. And after 5 minutes, voila! The clumped together pieces of DNA begin to appear.
So do you guys see like this little cloud thing forming? And what do you think that is?
A cloud.
Of what?
DNA.
So when you add
Yeah, so just pop it right in here.
The final product is encased in a small heart-shaped glass vial. It looks a little bit like the sea monkeys you may have raised as a kid.
If you imagine a piece of yarn, when you look at a piece of yarn, you see that in the detail there are small strands all swirled together, sort of. So that's what we were looking at in terms of DNA, the single strands all wound up together so that you could actually see it.
There's your DNA.
That is so gross.
All right, now I'm going to put this.
The DNA lesson comes from a grant from the University of Alaska Fairbanks. Lowe says the grant pays for all of the materials necessary for the program, which is in its second year. The exciting part for Lowe is to see the excitement in the students, a handful of whom just might end up being scientists themselves.
Well, that's my hope. When you have the opportunity to bring hands-on into any classroom, you see a special set of talents. Because not everyone is built to sit down and read a book or work problems. So, besides the activity, there may be a hint of what's next in science for me.
Or who knows, maybe someday one of these kids might become a forensic scientist and catch bad guys using DNA. In Homer, I'm Aaron Selbig.